All right, what's up, guys? My name is Zakir Wadood, and today I'll be talking about the buffer systems in the blood. Now, essentially, this is part of a broader topic known as acid-base balance, but in and of itself, it's not too much. Um, so, as always, I've highlighted what I think are the most high yield and important points in red. So, without further ado, let's get straight into it. So, first things first. As with anything, it's important to have a base in everything, pun intended. Sorry about that. I couldn't help myself. No, but in all seriousness, it's good to review the basics first. So it'll be great if you could show an understanding of the acid-base balance theory to the examiners. Like, for example, what is a Lewis base, what is the Bronsted theory, etc. So keeping it short and sweet, why is pH important, or why is the acid-base balance important? So, of course, as you all know, all the biological functions within our body are catalyzed by enzymes, and these said enzymes work best at a specific pH, and usually this pH is kept within short, tight, well-knit ranges, which is maintained by, you guessed it, the buffer systems of the body. So, first up, what is an acid? So, talking in plain terms, an acid is essentially nothing but a proton donor. So, something that releases H plus ions or protons, which would, of course, then mean the opposite is true for a base. I know, rocket science. So, something that essentially accepts a proton, or in other terms, something that can quickly remove an H plus ion or a proton from solution. So even in our blood, we have these type limits and restrictions regarding the pH. For example, in the case of arterial blood, we have a pH of approximately about 7.4, and this 0.00004 is just for illustrative purposes. Of course, you don't have to know it for the exam, but just wanted to show that what this is basically the proton concentration, meaning the amount of H plus ions or protons in a fully dissociated solution, which in this case is the blood. That would essentially be equivalent to a normal pH of 7.4. And in this case, it's 4 times 10 to the 10 to the negative sixth, or 4 micromoles, and slightly more acidic in the case of venous blood, around 7.35. And this owing to the fact that venous blood is, of course, as you know, less oxygenated and has more CO2 as well. Also, this has a lot of clinical significance to it. Um, as mentioned here, the lower limit to which a person can essentially still survive or live is about 6.8, and the upper limit is approximately about 8. Uh, and these are essentially the limits to which there wouldn't be any serious consequences. But of course, this changes for each individual, so it's not nothing written in stone. So take it with a grain of salt. But when it comes to urine, it's a little bit different. The range is a little bit more, say, flexible, ranging from 4.5 to around 8, and this changes again with the individual, the amount of hydration, and many other factors. It of course basically depends on the acid-base status of the ECF, or the extracellular fluid, for which the kidneys play a major role in correcting any abnormalities by either excreting any acids or any bases. So, as I mentioned in the beginning, this topic is essentially a subset of a much larger group of systems that essentially help in maintaining the balance of pH in the body, which will hopefully be covered by some of my other colleagues. So definitely stay tuned. Make sure to subscribe to our channel. But just to mention them, aside from the chemical acid-base buffer system, which is our topic today, we also have the respiratory system that plays quite a crucial role and function in regulating the removal of carbon dioxide. And typically does this through altering the pattern and the rate of breathing. And usually, compared to the other systems, this has more so of a quicker response. And a good example of this would essentially be something known as Kussmaul's respiration, which is a type of breathing pattern, heavy labored, quick breathing, seen, for example, diabetic ketoacidosis. Um, we also have the kidneys, which I spoke about briefly not too long ago. whose response is comparatively a little bit slower, usually takes from anywhere between hours to sometimes days. And it typically helps in maintaining the pH within these ranges, typically through the excretion of certain substances, usually certain metabolites or electrolytes, into the urine, and this would essentially result in either acidic or alkaline urine. And this by far is the most powerful of the three buffer systems. So back to our topic at hand, buffer systems in the blood. So kicking things off, we're going to start off with the bicarbonate buffer system. So the bicarbonate buffer system is essentially made up of a solution, which of course in this case is the blood, and it primarily consists of two main substances. So a weak acid, which in this case is the carbonic acid, H2CO3, and its conjugate base, which is the bicarbonate salt. And this is usually typically seen, for example, in the form of sodium bicarbonate. So the carbonic acid is continually formed in the body through this reaction, which I've listed here below. Carbon dioxide reacts with water in the presence of a very important enzyme, which you should know for the exam, called carbonic anhydrase, which turns this otherwise slow reaction into a much faster one. And this enzyme is actually also quite abundant in the walls of the lung alveoli, where carbon dioxide is typically released, and moreover, also in the renal tubular epithelial cells, where it tends to react with water to form carbonic acid again. And also in very much the same way, the bicarbonate salt, which predominantly occurs as I mentioned, sodium bicarbonate, since it's basically a strong base, it ionizes almost completely to form bicarbonate and sodium. And these constituents can then participate in what's sort of an equilibrium reaction, essentially going back and forth according to the acid-base requirements of the body. So that's essentially pretty much an overview of the um, bicarbonate buffer system. Next up, we have the phosphate buffer system. So this plays a huge important role in the renal tubular as well as the intercellular fluid buffering system, since it's found in very, very high concentrations there. The main components here are pretty self-explanatory. It's in the name itself. Um, and essentially, what these are a protonated and a deprotonated form of phosphate. So you have hydrogen phosphate as well as dihydrogen phosphate. The dihydrogen phosphate here, of course, being the acid, and the hydrogen phosphate being the conjugate base. The principle is essentially pretty much the same. The function of the buffer is to, of course, resist any change in pH. So when an acid or a base interacts with it, the corresponding component, if it's an acid, then the conjugate base, or vice versa, the base with the conjugate acid, it tries to react and neutralize that change. So as mentioned here, for example, we have HCl, which is of course a strong acid. When HCl basically reacts, it is then combated with the hydrogen phosphate, or sodium hydrogen phosphate to be more precise, which is base. And this then results in the formation of salt, as you can see here, NaCl, as well as the conjugate acid. So the decrease in pH, which would have otherwise happened if this buffer system were not in place, would be much, much less. So it tries to minimize or minimize the change in pH. I know this is rather a short topic, but there are a few other systems which will hopefully be covered by my colleagues. And finally, coming to the last and of course certainly not the least of the buffers, which are proteins as intercellular buffers. So as you know, proteins make up a large portion, a pretty good chunk of our blood, and it would be unfortunate if nature didn't find a way to incorporate that somehow into the buffer system. And of course, it did. As you know, hemoglobin, which is basically the protein inside red blood cells, which are responsible for carrying oxygen, also acts as a buffer for the majority of for the majority of chemical buffering actually takes place inside the cell as a result of the presence of this intercellular protein. Hemoglobin actually accounts for about 1/3 of the mass of an RBC, and likewise plays a huge role in the buffer system. during the whole process of the formation of carbonic acid from carbon dioxide and water, and its subsequent dissociation into bicarbonate and protons. The protons that are essentially freed or liberated in the reaction are buffered by the actual hemoglobin, which is then reduced by the dissociation of oxygen. And this then helps to maintain the pH within normal ranges, as shown or illustrated by this reaction right down here. And this process, of course, can go in both ways, and it essentially has to be able to go in both ways because, for example, in the pulmonary circulation or in the pulmonary capillaries, you have the formation of carbon dioxide back again from the carbonic acid because it is only in the form of carbon dioxide that it can be excreted or exhaled by the respiratory system to again help maintain the pH, keep it within those tight boundaries. And just a little trinket for you guys, a small flex, if you will, if you want in the exams. So the reason that the hemoglobin can essentially actually hold on to the liberated or freed protons is actually due to the presence of an amino acid residue, base actually, called histidine. So the presence of these histidyl residues on the alpha and beta chains of hemoglobin can essentially bind to the proton and hold on to it for as long as it needs to, and therefore and release it when the time comes in order to basically keep the pH regulated. And I know this was probably one of the shorter talks, but essentially this is one of the final questions. These three buffers systems of the blood. But as always, I hope you guys enjoyed. I wish you guys the best of luck in your exams, and please make sure to subscribe to our YouTube channel, and definitely stay tuned for more. And I'll see you guys in the next video.